also advise. I also advise the senior VP of research at Georgetown, and I chair the DASHU Innovative Design Scientific Working Group, which is a volunteer organization. So I'm going to talk about the definition of innovative designs, why innovative designs are important, evaluating their rigor and utility, optimal designs from the point of view of different stakeholders, tension between fostering innovation and ensuring rigor. And I'm going to try, if I can get through it now with the delays and realizing I needed to have time for questions, but I'm going to try to get through two contrasting examples of my experience with innovative designs that I have co-invented and am interested in having applied. And then I'll give possible conclusions. And regrettably, I don't know how much time there will be for audience discussion, but you can reach me at robert.beckman@georgetown.edu. Next slide, please. The standard clinical trial design evaluates one device or one drug and one hypothesis about safety and effectiveness in a single application. And it proceeds from the beginning to the end with no changes. And it often takes place in a centralized settings like hospitals and stuff like that. And innovative designs are many different approaches to varying that. So they're adaptive designs that learn as they go rather than staying the same, although you have to pre-specify the rules by which they will change. You can't make it up as you go along. There are master protocols that consider multiple drugs or multiple patient populations. Those are called umbrella trials and basket trials, respectively. Or multiple studies that begin and end at different times, like an assembly line with a single operational platform. Those are called platform trials. There's studies where the control group is derived wholly or in part from alternative data sources rather than concurrent patients. There are studies where measurements are done in the participant's home or by practitioners near their home or with wearable devices or Zoom. And these are called decentralized clinical trials. And there are also studies that take advantage of sophisticated mathematical modeling techniques, including the ever popular AI and machine learning and many other things. So it's a very broad category. Next slide, please. So innovative designs can potentially increase the efficiency of drug development as measured by the number of required trial participants, cost, and time. And many medical conditions don't have effective treatments, and innovative designs can bring more of these life-altering therapies to the market. Currently, it takes about a decade or so on average to develop a new drug, and the failure rate is very high. Innovative designs can potentially bring these drugs sooner. Innovative designs can be more patient-centered. Currently, it takes on the average of about $1 billion to develop a new drug, and the failure rate is high. Innovative designs could bring life-threatening therapy, life-altering therapies to the market at a lower cost. And here's a policy question, if we have time for discussion, which is, and I'm not a policy expert, I'm a oncologist and a mathematician, but currently many patients can't afford their medications. So the policy question is, if we were to reduce the cost of developing drugs with innovative designs, could the cost of medicines come down, or would that money just be pocketed by the companies? Next slide, please. So health authorities are faced with evaluating the benefit and risk of any proposed new therapy or device. And so they have to have information about the safety and effectiveness. And rigorous evidence of safety and effectiveness is necessary, or at least desirable, but health authorities can individualize their standard of rigor according to the judgment. So if you have a rare disease and there are 20 people in the world with this disease, and to do a statistically valid trial, you need 100 people, but those 100 people don't exist, then the health authority has to make various decisions about what they will accept. Typically, they'd like to see a well-defined hypothesis, well-defined study population, a low false positive rate, which is to say, if you put a drug that didn't work through this study design, how often would it falsely conclude that the drug works? That should be kept low. An accurate, unbiased estimate of the amount of clinical benefit. Study should be well conducted according to how it was written, with few exceptions. And the study design, especially if it's innovative, has to be prospectively designed. You can't go and look at the results and then change the design in order to make it a success. And for traditional designs, there are traditional established mathematical approaches for evaluating the rigor. But for innovative designs, that's often not the case. And often you can't even write down a mathematical equation, you have to simulate it on the computer. And also the definition of rigor may be a subject of debate. So next slide, please. So clinical trial designs may be evaluated on their efficiency. For example, how many true positives or drugs that work can you identify for a certain cost, a certain number of trial participants, while controlling the false positive rate? Or you can do it as, instead of true positives, net present value, which is a more economic thing that more accurate, but it requires more accurate input information. Innovative designs often have an advantage in efficiency, but there's a risk to the sponsor if we don't know whether the health authority considers the innovative design to be rigorous enough. Decision analysis is a formal way to analyze the net utility of the design to any stakeholder. And each stakeholder's value is placed on different outcomes that might result and is expressed in a utility function. So next slide, please. So different stakeholders may have different optimal study designs, and I was actually involved in a theoretical study where we designed different utility functions for different stakeholders, and we found that pharmaceutical or biotech companies, on average, the optimal trial design for them is smaller than for the optimal public health interest. And next slide, please. So there's this tension between innovation and rigor, and it's a challenge for health authorities because they want to encourage innovation, but they also need to ensure rigor. And the rigor of the innovative designs may be more difficult to evaluate. Patient groups often want innovation because they want to see available drugs come out sooner. But when they reflect, they also understand that they need to believe that if, let's say, the FDA says it works, that it works, you know, and if the rigor is reduced below a certain point, they can no longer have that assurance. So health authorities are faced with these conflicting pressures from different stakeholders, including patients and caregivers, pharmaceutical and biotech sponsors, payers, would-be innovators such as myself. Next slide, please. So I'm going to give you two examples of innovations that I'm interested in. One is basket trials. So multiple diseases or basket trials include multiple diseases or population groups that are traditionally considered distinct, but based on our increased scientific understanding, they have a key molecular characteristic or other physiologic characteristic that binds them together and actually makes them quite similar. And the premise is that this molecular subtype or other unifying characteristic is more fundamental than the traditional classification. So an example that's very common these days is in cancer, where it used to be that you had lung cancer, breast cancer, colon cancer, melanoma, all these different types of cancer. Turns out that that there may be a genetic mutation in a person with lung cancer, and that person may that may determine its sensitivity to drugs, and it may be more similar to a breast cancer patient with the same mutation than it is to other lung cancer patients. In fact, we now know lung cancer is a conglomeration of many molecularly different diseases. So you can pool these things completely, pool the data completely, or you can borrow information from one to the other. And this gives you the opportunity to study multiple indications, where an indication is an application of the drug. Indication would be lung cancer, breast cancer. Those would be, you can study several of those in one study and have a dramatic potential patient and resource savings. Next slide, please. So most of these basket trials have been, I'm sorry. So there are two phases of development roughly. There's the exploratory development where you get preliminary information about safety and efficacy. You determine the optimal dose and all this kind of stuff. And then confirmatory development are these large, very rigorous studies that lead to marketing approval from health authorities. So next slide, please. This is Willie Sutton, who was famous for robbing banks all across the United States, and he almost never got caught. And if he did, he escaped jail several times. And he was once asked, "Why do you rob banks?" And his answer was, "Because that's where the money is." And so the money in trying to improve the efficiency of drug development is in the confirmatory phase. That's where the money is spent. And that's why Willie Sutton is my favorite clinical trialist and mentor. So next slide, please. Basket trials have largely been exploratory, however. They've been single-arm trials without a control group, short-term primary endpoints like shrinking tumors. It's great to shrink a tumor, but it might come roaring back and kill the patient sooner. So survival is more important than shrinking tumors. And they've been opportunistic in nature. However, next slide, please. There have been exceptions. There have been about 6 or 7 approvals in the United States only of drugs that were really fantastic. These are drugs that already had a track record of being transformational, exceptional scientific evidence, and they were studied in patients with cancer patients with no other options at the end in the terminal phase. And so the FDA accepted these exploratory designs. The European counterpart did not. But I agree with this because of the situation. But if you wanted to generalize this to more drugs, you have to understand that most drugs are not transformational, but every biotech company founder with his or her drug thinks of his drug like their child and is not objective and believes their drug is transformational. Just like I assume my children are gifted, and numerous advances in medicine are actually due to just effective drugs. And short-term endpoints may not predict patient benefit. There are all sorts of biases that enter into a trial without a control group. And if you are trying to figure out if you can select patients who will benefit, you can't figure that out from a clinical single-arm trial because you can't distinguish whether your selection criteria are just picking patients who would do better anyway. These concerns were highlighted by the French National Health Authority in an article in Lancet Oncology, which is a very prestigious oncology journal. And the French National Health Authority basically said they don't want to pay for drugs that are made by these exploratory type basket trials. Next slide, please. So my volunteer group, which is now associated with DASHU, it was associated with DIA at the time, we wanted to develop a generalizable confirmatory basket trial design concept with statistical rigor that you could use for regular drugs you use with control groups. And this would increase and accelerate access to effective medicines for patients, provide sponsors with cost-effective ways to develop, provide health authorities with more robust packages for evaluation of benefit and risk. And again, most of the drug development resources are spent in the confirmatory phase. Next slide, please. So you might use these designs when you're able to have a control group, which is when there's a standard of care, so the control group gets standard of care. If there's no treatment, you're not going to give a dying cancer patient placebo. But if there's a standard of care that everybody gets, then you can add the experimental drug on top of it. When you need to measure time to event, things like survival, which really lead to patient benefit, you need randomized control. When you're looking at trying to enhance a drug that's already really good with it, adding something else to it, you need a control. The French National Health Authority article actually mentioned the designs that I'm going to very briefly discuss as potentially promising in the confirmatory space. Next slide, please. So this is the design, and clearly I'm not going to have time to discuss it, but it's kind of like a funnel. You start from the top and go to the bottom. And all these cartoon shapes are indications: lung cancer, breast cancer, colon cancer, et cetera. And you go through the design, and it weeds out the ones that are not promising, because if you're going to pool all these things together, they need to be the same. If half of them work and half of them don't, you could get false positives and false negatives from when you average it. And so you end up in this case with the two green indications, and you pool them, and you've now doubled your efficiency. You can basically evaluate these two indications for the price of one. However, next slide, please. This is a very complicated slide, and I'm just going to focus you on one tiny piece of it. So if you look at A, which is the false positive rate, and you look at the second brown row, and on the left, you see a basket, and it has some red marbles and some green marbles in it. And the green marbles are indications where the drug works, and the red marbles are indications where the drug doesn't work, known to a divine, supreme being. But the humans who designed this trial tried to pick 7 green marbles, and this is what they got: three green and 4 red. And then you go through this trial design, and it ends up approving 3 green marbles and 3 red marbles. Now, for cancer drugs, almost no cancer drug benefits every single patient. And certainly in lung cancer or whatever, colorectal cancer, that's not the case. So for conventional trials, if you have some green marbles in there, this is a true positive. But there are people who say that for a basket trial, if you have any red marbles in there, it's a false positive. And the reason is that people are uncomfortable actually pooling these things. The lung cancer patients, even though they're very different from each other, people got comfortable with that. But pooling a lung cancer patient with a breast cancer patient with a colon cancer patient, they're not comfortable with. And so next slide, please. So this is just saying that in a conventional trial with the lung cancer patients, they have all sorts of differences. Age is a big one. Other medical conditions, other drugs, even gender and race can lead to differences. And in this basket design, we're making the same assumptions, and we're actually making active efforts to reduce the heterogeneity, but it's still controversial, this idea that this could be a confirmatory study. Next slide, please. I would argue that if the scientific and medical justification for pooling is strong enough, there should be no need to control the false positives by the individual marbles. It should be the basket, just like a regular thing. But that's my perspective. But what we are doing now to support this argument is Satvik Narayana, who has kindly joined us, is sitting there, is actually looking at these traditional trials with traditional things like lung cancer that led to approval. And he's asking, how much heterogeneity is there really among these subgroups that we have come to accept? Because the basket trials should be allowed to have at least that much heterogeneity, in my opinion. Next slide, please. So the problem again here is perception. People are used to seeing this as a white vase against the black background. If someone comes around and says, well, actually, these are 2 silhouetted figures against a white background, people freak out, right? You know, they're used to seeing the white vase. They've been doing it for 30 years. They have departments arranged around it and so on. So next slide, please. So recognizing that some of those people who feel that way happen to be in highly empowered positions in health authorities, we developed another version of this, which controls the false positive rate by each of these little indications rather than the pool. And it still can give you a 75% increase in efficiency, a 40 to 75%. And what it does, it controls the false positive rate. The false positive rate is the same as if you had studied each of these individually in its own study. But there's some people who will probably say that's still not enough. So next slide, please. So there are lots of challenges with these innovative designs, and I give a whole hour-long lecture on challenges with basket trials. I'm going to skip from challenge number 1 to challenge number 14, which is epitomized by The Office here. And this is the standoff between health authorities and sponsors. So the health authority and someone like me is caught in the middle. So these things were invented starting in 2016. It's now 2023, and I published a lot of papers. I've talked with a lot of people. Some of them are ex-health authority people. Some of them are current health authority people, but they can only give their individual opinion, not the official opinion of the health authority. But the health authority people say to me, you know, we can't really give you a definitive answer about whether this design would be accepted until we have a sponsor come to us with a drug. Then we're going to invest the time. So then the sponsors call me and they hire me as a consultant, and they hear about the design and they get very excited. And then they say, so what's the regulatory risk that the health authority won't like this? And I say, well, I don't know, but they would be interested to talk with you about it. And they're sitting there, they're looking at a big payday of $1 billion if they're successful. They're going to invest $100 million or more in this trial. And I tell them, I don't know whether the FDA is going to like it or not, but the FDA would be happy to talk to them. And that's the end of my consulting gig, right? Because they're not going to take that risk when so much money is at stake. So there's a standoff between the health authorities and the sponsors. Now, the FDA has the PDUFA 6 pilot meeting program, where sponsors can meet with them to discuss it, but they can only do a few programs a year. It's a very competitive process to get into it. And furthermore, if someone like me invents something and isn't running a drug company, this idea cannot be considered normally unless I can get a drug company to take $100 million risk on it, right? So there's no way that I can have a meaningful conversation. I could talk to them. We have committees that I'm on that talks about this, but it's just talk. Next slide, please. So the standoff inhibits innovation, especially in the confirmatory phase where it might be needed most. Academics need to convince the sponsor to take a large financial risk. So the policy questions are, should the PDUFA 6 pilot program be expanded to cover more sponsor proposals, increase the size of the government to do that? You know, should we do that? Should sponsor payments subsidize conversations that they might have with academics? Because the FDA is funded by fees from sponsors who consider their drugs. I'm not going to pay them such a fee. Should the sponsors pay a little extra so that they have time to talk with me? I don't know. Well, I know what I think, but you know, I don't know what the right answer is. I'm obviously biased. Next slide, please. So the second example I'll go through very quickly is adaptive selection of a primary endpoint. And this is a happier story. This paper was published this year, and I'll go into it really fast. So this is for rare diseases. There are about 7,000 or 8,000 rare diseases that affect 30 million people in the United States. About half are children, and quite a few of them are progressive, debilitating, and lethal. So these are parents who are watching their children die slowly before their eyes. Within cancer, we're seeing many subgroups that are defined, molecularly defined, where cancer is now a collection of rare diseases as well. Next slide, please. Next slide, please. So these rare diseases, it's very tough to develop because they have, their natural history is not known. There's limited underlying information about the disease. There's a very small number of patients. There may not be any doctors in the world who see more than 2 or 3 of the patients. There's no research has been done before. And importantly, for what I'm going to describe here, people might not even know what to measure, which is called the endpoint. The primary endpoint is what people are measuring to determine if the drug works. They might not even know what a good primary endpoint is. So next slide, please. And also the health authorities have provided incentives for development in rare diseases, which are very good. but they're not necessarily enough on average to motivate sponsors to go into these rare diseases. Very small markets, very hard to develop. Next slide, please. So the FDA put out a very scientifically rational guidance where they said, well, if we don't know anything about rare diseases, then we just have to study the rare diseases natural history without therapy until we know these things. But what I can say is that I was in the pharmaceutical industry for 18 years, going to the R&D board, trying to get money for my drug, my project. And if I went with this, it would be a loser. If I went and I said, well, you can spend money for the next couple of years, we're not even going to use the company's drug at all. And then maybe you get small market from, you can start your study with the company's drug for a small market. After that, they would just tell me to shut up and they give the money to someone else, right? So how about patient advocacy groups and academics? Maybe, but it will take a while to do these adequately powered natural history studies, followed by adequately powered therapeutic studies. So if you were a parent with a child who was dying before your eyes, would you be enthusiastic about supporting that child to go on a study where they just watch the child die to follow the natural history? Maybe not. I wouldn't. So next slide, please. So we came up with a new design, which I won't go into in detail, but basically the design learns to select what to measure as it goes according to predefined rules so that you can do the natural history study and the therapeutic study simultaneously.
Sorry to interrupt, but we are almost run out of time. So if you want to start wrapping up.
I'm about to do that. So yes, so in any case, it's called the informational design. We'll skip the next slide, which is a diagram of it. We can skip that. Next slide, please. And so this had a very different outcome. So I presented this, and I'm going to get to the role of patient advocacy groups here. I presented this at a session at the DIA meeting, which was about how innovative designs could benefit patients. And I believe there were patient advocates there. Anyway, an audience member stood up and said, well, when is this going to be applied? It's very exciting. And I said, well, I have absolutely no idea. All I can do is publish in the journal and hope that the FDA or the EMA will, will say something good about it so that companies would actually use it. And so the second person stands up and says, Dr. Beckman, you're being evasive. Tell us when you're going to apply this. And so I said, well, maybe there's someone from the FDA who could help me with this because I'm not being evasive. I honestly don't know, And woman stands up and she happens to be the director of the biostatistics office at the Center for Drug Evaluation and Research at the FDA. And she says, you have an invitation to talk with us. So I talked with them and we're now working together with them. They had some questions. We're trying to answer their questions. And this is what, if the FDA and the EMA had the capacity to do this, the problem is that I'm not the only one, right? So there's lots of people like me with lots of ideas. And, some of us have good ideas and some of us have bad ideas, and we all think our ideas are good. So if they're going to listen to me, they have to listen to 100 other people. And maybe out of the 100 of us, there's only 2 of us that are really worth listening to. And it takes a lot of resources. So this is a public, I would say, a public policy question. And I don't know if we have any time for discussion, but that's pretty much it. Next slide just sort of summarizes, I think, what I said. Yeah, the health authorities are trying to promote innovation, but it's slow to take hold. How can we encourage innovation and still maintain rigor? Understanding that you didn't see any of the technical details. I may be completely out of my mind. And if I'm not, there's someone else who is. They're going to have to evaluate everybody if they talk to me. So anyway, is there time for one question or no? Okay. We can have one question here. Okay. Anyone have a question? Anyone have a question on the virtual?
No, sorry.
Oh, I was going to say, if anyone has questions later, since I didn't allow time for questions, please email me at robert.beckman@georgetown.edu. But I'd like to, if there are public policy people listening, this is something that I know nothing about public policy, and I recognize that this is a very important public policy issue, I think.
Thank you for your interesting presentation, and I'm sure people will contact you with follow-ups and questions.
Okay.